I'm going to demonstrate the use of triple sugar iron agar in the laboratory. Usually we use this specific type of 3-sugar media when we're trying to differentiate among the members of the Enterobacteriaceae, especially if we're trying to differentiate from other gram-negative bacteria like Pseudomonas aeruginosa. Now notice that there's parts to this media. There's a slant portion and there is a butt portion, and we're going to inoculate both. The slant is going to remain aerobic, but the butt is going to be the anaerobic part of the experiment. Now, the way we're going to inoculate this is by using both parts of the tube. And we're going to use a loop and a needle for the inoculation process. I prefer to use the loop to place the bacteria on the slant portion, and then the needle to stab the butt portion of the tube. I'm going to just do a zigzag on the surface of the slant. I'm going to incinerate my loop, and I'm also going to incinerate my needle so that we actually can use proper aseptic technique. Now in this specific exercise, we ask that you inoculate with the needle. You do the stabbing multiple times. So if you have any frustrations, you can probably take them out on the stab. So I'm gonna go 1, twice, 3 times. And this is ready for incubation at 37 degrees for 48 hours. We're going to look for a color change. There's a pH indicator in the media that is going to change to yellow if it's acidic and to pink if it is alkaline. Now, here you have results after 48 hours. For E. coli, notice that both the slant and the butt are yellow. And I want to point out that there is a bubble. This is going to be because there was gas production and the gas was trapped in the tube. Here you have a slant that's pink and there was no change in color in the butt. I'm going to talk about why that is. So the pink color indicates alkaline breakdown of peptone in the aerobic area, and there was no fermentation in the anaerobic area. Here you have alkaline results on the slant. When the organisms run out of glucose that can break down peptone, they'll do so, and the color will go from yellow to pink. At the bottom, you can see the yellow color for fermentation in the butt or anaerobic portion of the slant. It is completely possible to have pink at the bottom if the organisms are able to live in an anaerobic environment.